Okay, hello everyone, and welcome back to another CEN seminar. Today we have a very exciting session. We have Michael Thomas, who needs no introduction really, but I will give a little introduction for him. So Michael is a professor of cognitive neuroscience at Birkbeck College, University of London, and since 2010, Michael has been director of the Centre for Educational Neuroscience. We are a cross-institutional research centre which aims to further translational research between neuroscience and education and establish new transdisciplinary accounts in the learning sciences. You all know this, I'm sure. In 2003, Michael also established the Developmental Neurocognition Laboratory within the Centre for Brain and Cognitive Development. And the focus of his lab is to use multidisciplinary methods to understand the brain and cognitive bases of cognitive variability, including a focus on neurodevelopmental conditions. And Michael, I will say, is also the pioneer, let's say, of coming up with kind of multi-level explanations for things and trying to connect different levels of explanation. So today we have a topic of that type, I would imagine, which is heritability is not destiny. So I will hand over to Michael.
Thanks very much, Astrid. Thanks so much for the introduction. I will endeavor to share some slides. Just bear with me. I've got zillions of windows open, and one of them I need to share. One of them.
Don't worry, Michael. I feel like I should be doing a little dance to keep people entertained.
If you have buffs, that would be, of course, terrific.
Well done. Well, we can see them and they're moving.
Excellent. Okay, sorry about that. So, yesterday's title is called Heritability is Not Destiny, and I hope that title will become self-explanatory as we go through. So this talk is intended as a sort of companion piece to a presentation that Emma Meeburn gave 2 weeks ago to this group on genomics and education. For those who didn't attend or don't recall the contents, here's a brief recap. The argument is that genomics is relevant to education because 50% of the educational gaps that we see in outcomes are correlated with genetic variation. Now, previously, that was established by twin studies and assessing levels of heritability. But there's now a new focus using genomics and focusing directly on DNA variation and findings stemming from genome-wide association studies. And those are very large-scale studies. The effects on educational phenotypes like how you perform in exams or the very rough and ready metric of how many years you were in the education system. There are very, very tiny effects can be traced to individual letters of DNA variation, of variations in DNA sequence. And it appears that there are thousands and thousands of such variants that seem to predict variation in high-level phenotypes such as educational achievement and educational attainment. So the main finding that comes from those studies is to the extent that genetic variation is predicting variation in genetic outcome, it's highly polygenic in the general case. What is notable, however, is that these large-scale studies, so I think the most recent one had 3 million participants in it, 3 million adults who reported the educational level that they attained and gave their DNA. was that you can come up with a recipe or a formula that anyone can plug their own DNA into, and you can then generate a prediction for your DNA about what educational attainment your DNA says you are likely to achieve. So that's kind of a step forward. Much of the previous study of genetic influence on individual variability only spoke to the group level, but now with the arrival of genomics, we have prediction pertinent to the level of the individual. So that is an example of data coming from the most recent genome-wide study of educational attainment. And what you're looking at on the left-hand side is like a scatter diagram of, I guess, like all 3 million adults, polygenic index, what the formula generated as a prediction of their educational attainment from their DNA. And then you're seeing on the y-axis how many years they actually went in education. And you can see the regression slope there. So the relationship is explaining 16% of the variance. Directly from measuring someone's DNA, you can predict 16% of the variance in how well they're doing education. If you're a glass half empty person, I guess you say that's 84% of the variance unexplained. But what's notable is on the right-hand side, I'm showing the predictive power of some of the main socio-demographic variables that are often used by policymakers to examine how education systems are working. Things like parental education and family income. And now these studies, genome-wide association studies, had to get quite large before they started generating predictive power. That's because of the small size of the genetic effect. So this predictive power of 16%, you're only getting that with a sample of 3 million adults. But because all of a sudden you're getting predictive power up there on the level of socio-demographic variables, that has caused policymakers all of a sudden to think, well, should we be beginning to pay attention to educational genomics and what might its implications be? So I'll give an example of that interest. So the UK Department for Education, they periodically produce a document called Areas of Interest, questions they are interested in that are pertinent to the current government's policy interests. So at the moment, the current government has several missions. One of those is an opportunity mission to reduce barriers to opportunity in society. And one of the mission pillars is called the Best Start in Life. And this document was requesting from researchers information about genomics, asking what are the links between genomics and SEND conditions, special educational needs and disabilities, and how might we best prepare educators, care providers, parents, and children for increased genomics data. And there's been a sequence of reports. So there's one from 2022, Genomics Beyond Health, one from 2024, Genomics Implications for Education, and a couple of reports stemming from the Nuffield Foundation. One of them, the lead author was indeed Emma Meeburn, Navigating Genomics and Education, Insights, Opportunities and Challenges, prepared while Emma was there on secondment. So interest amongst policymakers now in the relevance of educational genomics. I'm going to say very broadly, those reports have taken the view that the application of genomics to education is in the future, that it's not yet ready, powerful enough to be practically applied to education. And the reasons for that, I hope, will become apparent as we go through this presentation. But because in the area of genomics, we now have not just DNA being collected and analyzed by researchers, not just within the health system, but we also have direct to consumer companies, and you can send your DNA off, and they will send back information about your ancestry and your health risk. And in principle, as of now, because the genome-wide association study data is generally publicly available, including for educational attainment, you can send off your DNA and get a prediction for yourself or your family about how well you are likely to perform in education. Now, even if we can view it as being premature to practically apply this information to the education system, we still are presented potentially with this kind of challenge. You are a teacher at the school gate, an irate parent comes up to you at the end of the school day, takes you to one side and says, I sent my child's DNA off to one of these direct-to-consumer genetic testing services, and it came back and it told me that my child is in the top 10% for educational attainment, but he seems to be in the bottom half of your class. You're messing up. So even if we can conclude that it's too early to be using genomic data in education, we still may be faced with this kind of challenge. And the question is, how should a teacher ask that question, respond at the current time? And I hope to get to, by the end of this presentation, what I think an appropriate response would be for that teacher. Okay, so I'm going to cover 3 topics today. First of all, I'm going to suggest if we're thinking about the application of educational genomics, we need to distinguish between research and translational aims, because I think those 2 different aims have different emphases on what you need from the science. Secondly, I'm going to suggest that we need an understanding of developmental mechanism in order to understand how things might turn out differently. That our current data is reporting how things currently are, but if we want to know how things could be different, we do need to understand something about mechanism. And in that section, I'll be using a simple computational model to generate some intuitions about in biological systems, in child development, how could things turn out differently? And then I'll finish off by speculating a bit about future directions of applied educational genomics. Okay, so let's start off thinking about educational genomics for research. And I refer you mainly back 2 weeks ago to Emma's presentation. She went into a lot of detail about that. But if we think about the broad aim of a research approach, the goal is to shed light on the causes of individual differences in a phenotype in a given population. So for education, where in this population do the gaps in educational outcomes appear to be coming from? And that involves collecting lots of data, genomic data, phenotypic data, measure educational outcomes, look at measures of the environment, of parental education, family income, quality of the schools, these kinds of sorts of data to understand causes in your current sample. And you use various sophisticated statistical methods and various assumptions to give you potentially insights into what are producing those differences. So this is an example. I went to a very sophisticated lecture on kind of research genomics on behavioral phenotypes, and I scribbled down what kind of moves do you make if you're doing research in this area. Let's say you've got some new behavior, you're interested in exploring it. This behavior shows some variation. How, from a research perspective, would you go about investigating it in a research paradigm? Well, the first thing you do is you would establish the potential heritability of your phenotype. And you generally use twin studies for that. You need to know if there's a genetic influence on variation in the behavior before you even bother collecting DNA and looking at genomics. If there's no genetic effect, there's no point in doing the genomics. After that, you might then do a genome-wide study if you can get enough people in your sample showing variation in the behavior. And then you would generate your polygenic index, go have a look at the letters of DNA that are varying see how big the variations are, do various tests of those. You might explore those letters of DNA that are varying, that the DNA sequences fall in certain genes or regulate certain genes. What do we think those genes are doing? If you're looking at a behavioral phenotype, you're hoping some of those genes are going to be expressed in the brain or certain kinds of brain tissues. And after that, you might think, well, my polygenic index for this behavior, does it correlate with other polygenic indices for things like cognition or ADHD or educational attainment? So you're kind of looking for the overlap to get some, hopefully, insight into the causes, genetic causes of variation. And then you might say these genes that look like they're varying, do we see in rare conditions sort of mutations producing big differences in this particular behavior? And we might correlate our polygenic index to PGIs for other phenotypes like brain size or cortical thickness or these kinds of things. This is the general way we go about doing research in this area. And some of the sophisticated techniques we might use are, so we've got our polygenic index here, and you can see big variation. Now, these type of indices are often normally distributed. Most people get the average score, and a few people have it the extreme low, and a few people have extremely high. If you're investigating how the genetic influence is working, you can just compare people in the tails, the lowest ones and the upper ones. By doing that, you're exaggerating the genetic influence in your sample, which makes it easier to investigate. And you can do other sophisticated things. So one of the very impressive things that Emma talked about a couple of weeks ago is comparing the parental polygenic index to the child's parental polygenic index. Why would you want to do that? Well, we know if a parent is highly educated, maybe they're highly educated partly because they've got good genes for education, whatever they are. And they might pass on some of those genes to their child, but they are also likely to create a certain kind of parenting or environment for their children to grow up in. So you'll get some similarity between the parental phenotype and the child phenotype, but you won't know whether it's direct genetic transmission or whether it's due to parenting. And probably the parenting part of it is more relevant if you want to make policy advice or generate guidelines about enriched parenting experiences for education. And you can use the polygenic index of the parent to control for the sort of what we call a parental nurture effect, that kind of parenting they give by virtue of their genes. So these are sophisticated ways to untangle the causality that's producing individual differences. Okay, if we switch to educational genomics for translation, there's a different kind of aim here. We now need to understand what range of alternative phenotypic outcomes are possible if you have a population with a similar distribution of genotypes. If you take that population with those genotypes and raise it in a different sort of physical environment, maybe of diet or health, or a social environment, or an environment with different educational conditions, what kind of range of outcomes could you get instead? Because then those physical, social, educational changes might then be the target of different policies. So that's what we need for translation, to understand what you might call counterfactuals. How can things possibly turn out differently under different circumstances? And for us in educational context, we're interested in, what policy changes could you make to produce fewer gaps, narrower gaps in educational attainment in your population. So interesting, if you're doing that, probably you no longer want to focus at the extremes of this distribution. Really what you're interested in is, for a given genotype, for a given polygenic score, what is the maximum range of different outcomes I can get? Because that's pointing to the fact that if we control for your polygenic index, presumably it's environment that's causing all these differences. So that probably leads you to look at the middle of the distribution, where we see the maximum variation in outcome for the largest number, presumably for the environmental cause. So that will be where you would go investigating, well, what are the social influences, educational influences, or physical influences that are producing that wide range of variation in the middle of the distribution. I'm going to argue that if you want to examine counterfactuals, how things could work out differently, you need some degree of understanding of mechanism. What is causing the differences? You need that to understand how the system will behave when the environmental variables and influences are altered. Now, understanding a mechanism, that is the biggest challenge that genomics faces right now. I would say those genome-wide association studies that you've been looking at and hearing about, they are mainly a data-driven approach. They're not theoretically motivated. And we get these observations about the associations between genetic variation and phenotypic variation, and only afterwards are we trying to figure out how they might be caused. So the challenges in understanding mechanism are multiple. One of them is biological complexity. To give an example, a synapse is a tiny structure involved in the communication between neurons at a micro scale. And if you pull apart a synapse, you find out that it has like 2,000 proteins influencing its function. And we know that one gene codes for one protein. So that just shows you the scale. Even just getting to the synapse is very, very complicated, let alone whole brain function. We know that behavior is generated by the whole brain in the body. We know that there's been an extended developmental trajectory involving interactions of the individual with their physical and social environment and educational environment. And we know that often that is proactive. It's guided by the child. It's not just a passive reception. And we know that of the environmental influences, they too are very complex. They're nested. They involve influences of the family, the school, the community, national factors, and so forth. So understanding the mechanisms is going to be very complicated. So this is a barrier to translation. If we want to know counterfactuals, we have to understand mechanism. Mechanism is very complicated, but it's not the only barrier to translation. Here are some other ones. Emma talked also about concerns around ethics, data privacy, consent to give genetic samples, inequities that arise from unrepresentative samples that the genome-wide association studies have been based on, and the generation of polygenic indices. There is another problem that our current polygenic indices, even if that education one is predicting 16% of the variance in outcome, it is insufficiently powerful to be useful at the individual level. And it turns out a lot of these polygenic indices for different kinds of traits seem to correlate with each other. That is to say, they're not very specific to particular traits. It's sort of a double whammy, not powerful enough, not specific enough. And then in education, there is a particular problem with a fear around determinism. Now, I'm going to focus on that fear of determinism in the middle section of this talk. So fear of determinism, I mentioned a report that came out of the Department for Education in 2024, and this did pick up and emphasize this issue around determinism and the view that the general public and teachers are quite likely to view the use of genetics within a deterministic framework, as if something is predicted by genetics, it's an inevitable outcome. This is partly linked to a problematic past in the association of genetics with certain extremist views or right-wing views, however you want to characterize it. Why it's problematic is, first of all, it can become self-fulfilling. If you believe a polygenic index score points to an inevitable future, it's like assigning a label. In education, there is like a well-understood labeling theory that by labeling someone as highly able or having low ability, you will act in a way that actually causes that outcome. It's also problematic in the sense if you think the outcomes are inevitable, you would think, well, why would I change policy? I mean, this is more broad. It would work for, say, poverty as well, or any of these structural aspects of society. If you think they're the deterministic consequences of genetic variation, you will just give up on the idea that you can use policy to change it. So that's an abstract notion. Here's an example. This is a blog from a commentator who was involved in setting up free schools in the UK back in this is a blog from 2017. Now, I will admit this is quite a controversial blog, and I'm just using it as an example to demonstrate this type of thinking. I would say the science reported in it is not inaccurate, but I would probably disagree with the interpretation of that. But this is someone who's involved in setting up schools and realizing after some time that however they seem to structure their new schools, they didn't seem to close the gaps in educational outcomes. And this is a blog suggesting there might be limits to what schools can achieve in narrowing gaps, and suggesting this is because a lot of school achievement is related to intelligence, and intelligence is highly heritable, and therefore not amenable to be changed by what a school is doing. So that's a clear demonstration of a deterministic view that a highly heritable trait, that is, genetic variation is strongly predictive in phenotypic outcome, implies some kind of destiny in outcome. Okay, so a little technical bit now. I'm going to use some computational modeling. This is one method to focus on mechanisms. So what we want in this context is we're not just interested in the mechanisms in the brain of a single child. We're interested in studying development in a large group of children, in a population, indeed. So of course, we're going to have to make simplifications. We don't know how to build a model of a whole child. We're just going to focus on a single developmental mechanism. Here it's going to be an artificial neural network representing an individual child. And each one of these modeled children is going to go through a developmental process. And we'll build three assumptions into our model. The first one is that any behavior shown by the child is the outcome of experience-dependent developmental systems that have plasticity, that they change and improve as a consequence of experience. The second is that polygenic influences, or that DNA varying, is acting on the neurocomputational parameters of these systems. This is sort of an information theory, information processing view of what the brain is doing. And those genes are making these neural networks more plastic, or more able to learn complex ideas, less noisy, these kinds of things. And then we'll assume that the environment is operating mainly by influencing levels of cognitive stimulation, how rich the developmental environment is that the child is raised in. Okay, and our goal in using the model is to directly test the idea of whether heritability is destiny. Okay, are highly heritable traits inevitable outcomes? So we'll have a developmental mechanism. It's going to interact with the environment. It'll go through a developmental process. And as a consequence, there'll be some improvement in behavior over time as the model child develops. What's going on inside our mechanism? Well, there's this artificial neural network, and it's processing properties, so like how many neurons there are in there, or the levels, the plasticity of connections, these kind of things. They are going to be influenced by an artificial genome, which has lots of genes on it and lots of variation. And in principle, yeah, you could correlate variation in one of the genes because it contributes to the variation in the properties of the network, and that goes through a developmental process which ultimately produces behavior. You could do those kind of correlations between genetic variation at the lowest level and behavioral outcome. And what we're going to do now is to say, okay, that's an individual, but we're going to have a population of those, and we're going to have variation at the genetic level, producing networks with different learning abilities, and variation in the richness of the environment to which they're exposed. The genetic level then allows us to do two other things. we can we can generate identical twins and non-identical twins. We can generate a standard genetically sensitive twin design that allows us to estimate heritability. Or we could generate artificial polygenic indices and see if we can use direct genetic variation to predict differences in outcome. Okay, so what we'll do is we'll have a population of kids, and early on, some of them are developing quickly, some of them, most of them average, some of them a bit more slow. Come back a couple of years later, they're getting better. Some kids maybe have changed position, some kids have slowed down. But you'll still get a normal distribution, and the whole distribution will improve over time. Okay, so we need to manipulate heritability. We're going to create populations where the differences in behavior are either mainly from variations in genetic influence, it's a high heritability population, or mainly due to differences in variation in the environment, that's low heritability. So this is some results from the model, and you're looking at, this is like early childhood, let's say, how along the bottom is the performance level and how many kids are at various different levels. And the difference in the correlation between the MZ twins and the DZ twins is your measure of heritability. So the dark blue, the low heritability, the numbers don't really matter, 0.47, but the high heritability, 0.57, it's a high heritability population. In this case, you can see the light blue has a wider variation, and that's because they've got wider variation in genetic abilities. Okay, so means are about the same. For that population, for those two populations, they're generally raised in nice environments that aren't, not massive variation in the environment, or the environments are reasonably kind and enriched. But we can do the same. We can raise those populations in environments which can have much more extreme variation. So in principle, you could have quite impoverished environments or deprived environments. Okay, at that point in development, I'm going to say stop. Let's do an intervention. The intervention will be universal cognitive enrichment. Every child gets the best possible experience that they could. And then we're going to test, is that intervention, is it going to be less effective in the high heritability populations, the ones where genetic differences are mainly responsible for producing differences in behavior? I'm showing you the low heritability, the blue, that's the under normal circumstances. Red is the counterfactual, what happens when I intervene. And you can see I've followed up, a little bit later in childhood, and everyone has improved. What do we think is going to happen in the high heritability population? If it's destiny, we're just not going to find the same kind of change. What do we find? Oh, actually, it's exactly the same size change, actually a tiny bit bigger. Heritability here is not restricting your ability to improve these kids. You could say, well, okay, you've accelerated everyone a little bit along development. Okay, but when we get to the adult outcome, that's really where the genetics cashes out, right? That's where in the high heritability population, you won't be able to close those gaps. So this being a model, we'll come back in adulthood and see what's happened. And, we find exactly the same thing. Everyone is improved, but you're still able to improve both populations, high heritability and low heritability, to the same extent. We can do the same exercise here where we've got potentially much more variable environments. You'll see that in the blue, much more variation in the outcome. You improve everything for everyone, you have a much bigger impact now. So the intervention is having a much bigger effect because you're helping all those kids who are performing poorly because they were deprived. What happens in the high heritability population? You get the same mean size improvement. Now, I will say there's more variability still in the red population, the high heritability population, than in the low heritability. But just wait a bit, come to the adult outcomes, and that goes away. And again, same pattern. Equal facility to improve the outcomes in low heritability and high heritability traits. So What that's telling you is the systems producing those behaviors are equally malleable. And that's because levels of heritability do not tell you about the plasticity or malleability of the underlying system. In all these cases, these model children have developmental plasticity. So they all respond to intervention. And the size of the intervention effect, that depended on how much the impoverished environment was holding individuals back. It wasn't to do with the genetics. That's all doing heritability and twin studies, but we're kind of interested in the polygenic index, this kind of thing. So we can generate a polygenic index for our model populations. This is a low heritability and high heritability, normally distributed polygenic indices, as you'd expect. Low heritability, it's explaining 16% of the variance, and then it goes up to like 40% in the high heritability population. That's what we mean by heritability, that the genetic variation is explaining more of the variability of outcome. But this is just reporting what is. What could be? What if we took individuals from any part of the distribution of this polygenic index and saw what range of possible developmental outcomes could you get if I raised you in different kinds of environments? So we can explore that by doing that, taking a genotype and just raising them in lots of different environments. And what I'm showing you is drawing individuals from the low heritability or the high heritability population and showing the possible developmental outcomes. I picked out, random 2 individuals from each of decile, just showing random cases of what possible developmental outcomes could that genotype produce if raised in different environments. And what you're seeing is huge range of developmental outcomes are possible from all positions on the polygenic index. Yes, there's a broad pattern that on the left-hand side, the outcomes are a bit lower than on the right-hand side. But you're seeing big effects of the environment, effectively, irrespective of the polygenic index. So that's just exemplifying it. Really, we should put some numbers on that. So what I'm showing here is either the low heritability population or the high heritability population. How much variance is the polygenic index predicting? So we can see in the red there, it's like, 0.2 and whatever. And then in this case, obviously in the high heritability, it predicts more. And then how much is being predicted by the environmental variable, how rich the environment is. And then are these effects independent and additive, or do they interact with each other? So does it matter where on the polygenic index distribution you are for the effects of the environment? In the low heritability population, doesn't matter at all. In the high heritability population, it matters a tiny bit. But broadly, what you're seeing is big effects of genetics, big effects of the environment, but they're sort of operating independently. Now, that's a model, of course. You can go look at real data, and you kind of find the same thing. Big effects of genetics, big effects of the environment. I'm showing you data from a Norwegian population sample here, where they looked at 7 different environmental variables and could only find an interaction between genetic and environmental effects in 1 out of 7. And even that was pretty small. And that's unusual. Most European samples and the UK data I've seen, there are no gene environment interactions. That is to say, you get the same effect of manipulating the environment irrespective of the genotype. As an aside, slightly different patterns from America, where there's a lower social safety net. Some arguments that very deprived environments can start to attenuate genetic effects. So the take home from this, plasticity allows lots of developmental outcomes for all genotypes. In both models and real data, gene-environment interactions for education are small or absent. What does that mean for the teacher? It means, look, children in your class are going to differ in their progress because of genetics. But you have equal facility to help all these children. They will improve by the same amount. And if you wanted to narrow the gaps between children, you just help the lower achieving kids, the lower performing kids more, and then you will narrow the distribution. So this is a message of agency to teachers, even in the face of these sort of genetic effects. So I'll finish off the modeling bit with a puzzle. So if you want to do that, if you want to narrow the gaps, what you do is you give more help to children lower in the distribution. Let's say that the bottom 50% or the bottom 20% or something, our special educational needs and disabilities system and our free school meal system does that. They allocate more resources to people lower down the distribution. But here's the puzzle. How should you define, say in this case, the bottom half? According to what? Should it be the current achievement of the child, their behavior? Should it be their genetic potential, for instance, from the polygenic index? Or should it be according to your estimate of their current level of cognitive enrichment, say as indexed like a metric like free school meal status in the UK? Which of those would close the achievement gaps the most? Again, we're into counterfactuals, right? Well, in the model, because you can explore these different possibilities, you can test these, you can compare. And the answer is, actually, there'd be a drum roll, maybe a drum roll here. It's current achievement. Current achievement is the best way to narrow the gaps if you're doing differential interventions. And that is because it is wrapping together both of the other two, both of the genetic influences and the prior cognitive enrichment. And that is why it's a more powerful predictor to help you narrow gaps. Okay, but this is a model, so we have to be cautious in that. But I'm just trying to use it to demonstrate that you need to understand developmental mechanisms to explore the counterfactuals that allow you to do translation in educational genomics. And the fact that we understand that cognitive systems, brain systems are plastic suggests that these deterministic interpretations of heritability, of polygenic indexes and heritability and so forth, that's unwarranted interpretation. Okay, so shorter last part before I wrap up. Future applications of genomics in education. Now that education is generally in this area following in the wake of innovations in health studies. And there are large-scale health studies now ongoing that are recruiting like millions of participants, collecting genomic information. And what is notable is the latest versions of these are offering to give back genetic information to their participants. So this is kind of a heads up about, if this is 10, 20 years in the future, what might this look like for education? So I'm showing you a screenshot from a seminar that took place in the summer, hosted by the PET organization, which was just talking about these new health cohorts that were being set up. And this is one being run in America. It's called All of Us. And this slide is just demonstrating the types of genetic information that this new study is giving back to its participants. But also, they're doing research around that. So what are people interested in about their own genetics? And how are they using it and how is it affecting them? And I just give this as an example. So what information did they give back? They gave back people information about their ancestry. And 220,000 of the participants accessed that data. Even more accessed kind of gee whiz data about you're likely to have sticky earwax or bitter taste perception or strange things like that. About half as many were interested in genetic variants related to disease risk, but it turned out to be only relevant for about 3% of the sample. And about the same number were interested in directly medically relevant genetics around the kinds of medicines that you might have a reaction to. Okay, so interesting the kinds of things that genetic information is being fed back, but people are sort of more interested in the gee whiz part than they are in the direct medical part. So at the Center for Educational Neuroscience, we have convened the Genomics Working Group, which is being led by Emma, and here are our glorious members. Mojtaba Sultanu, Yulia Kovas, and Joe Van Herwegen, Tim Morris, Emma Meeburn. I'll just put up a paper by Tim Morris because I mentioned early on that our current polygenic indices, including for educational attainment, don't seem to be powerful enough to strongly predict what's going to happen to children at the individual level. This is a paper that Tim published with Neil Davies and George Davey-Smith, specifically running those analyses and saying that the current polygenic indices are just not powerful enough to be used at the individual level. So what have we discussed in this group? What positions are we working towards? Well, several of them. This is the I think 4 pathways we're sort of picking out about how we see the application going. And we think the first and most likely pathway is really quite a narrow group of children who've already been clinically sort of phenotyped and genotyped, and it's relevant to their health condition. So these are effectively health-linked genomic data, but some of that is going to be relevant for education as well. So that's closer to the health end of things for a small number of children. The second broader notion will be the one picked up in that Department for Education report, probably around SEND, just beginning to think about, could we have a soft, early available marker for the potential for special educational needs and disabilities conditions, to inform parents, to help early as practitioners, teachers, and so forth. We're still up against the fact that the polygenic indices we have are not really powerful enough to do that. And there are some negative sides of that. It's a bit like a medicalization of SEN conditions, and we risk deterministic interpretations around that. And probably at the moment, those polygenic indices are not really fit for purpose. Really, we want them to predict educational need, not to be focused on diagnostic category. Because, I mean, this is a change in that field, a de-emphasis on labeling and categorization, and more about identifying, what is this child likely to need in the classroom? So that is still down the road. There is a possibility of personalized education. This is often raised by behavioral genetics people speculating about education. At the moment, we await more powerful and more specific polygenic indices to allow us to do that. The lack of gene-environment interactions probably at the moment is telling us that you're only going to use that polygenic index for identification. It's not going to tell you what you should be doing with that child, because the lack of a gene-environment interaction is telling you that, the environment works the same way for all these children. So just use your established best practice educational methods. And if you're going to use the genetics, if it ever gets that powerful, then that's just for identification, and then use your best techniques. Again, concerns around deterministic interpretations if you use genetics in that way. And then the last possibility is sort of stepping back a bit and thinking, well, maybe we want to use genomics mainly for research aims. It's less contentious there. We'll stick probably more at the group level, but now we can use this as an additional variable to help us investigate policy relevant issues about, say, the effects of policy changes on distributions in populations. For example, it's often taken that if you increase the heritability of say educational outcomes in a population, that is because you've improved and equalized the environment for everyone. Or I alluded to earlier, you can be using your sophisticated genomic methods to try and tease apart direct genetic inheritance between parents and children versus influencing going via parenting. So here's my conclusion. Genomics not currently powerful enough in its predictions to be of practical use at the individual level in education. A lack of mechanistic understanding limits a broader translational application. Deterministic interpretations are likely misplaced because they don't take into account developmental plasticity. And The current evidence, at least for the phenotypes we have in education, is that children seem to largely benefit from the environment in the same way. However, part of this is PR, because if you believe in determinism, it can become self-fulfilling even if it isn't real. So there's an important aspect about messaging to help understand that the heritability is not destiny, hence the title of this talk. Okay, so we'll finish off here. We got the angry parent. How do we respond to that parent? Polygenic index is in the top 10%. Child is in the bottom half. Well, let's go back to the actual polygenic index. Let's have a look. That's the top 10%. The red dotted line is the population average. Yeah, there are going to be some kids like that in the top 10% who end up in the bottom half. Here's what we say. I'm afraid these tests are currently not very accurate. They are fun though, aren't they? But we should definitely work together to see how we can support your child's progress. Super, thanks very much for your attention, and thanks to everyone who has contributed to the material in this talk.
Absolutely fantastic, Michael. I mean, thank you so much for such a comprehensive and, such a thought-provoking talk. So we have got a few interesting comments and questions in the chat already. I will sneak in with a quick one. So I don't think any of these questions are sort of softball questions, so I haven't got an easy one to start you off with, unfortunately. But you kind of came back to this a bit at the end, but I thought it was really interesting how early in the talk you kind of defined genomics for research and genomics for translation. And I just wondered if you had any thoughts on any sort of implications of those distinctions. Are you suggesting that we should be more specific about what a piece of work is trying to achieve, or, should we be sort of marrying these goals up better in some way? I don't know if you've thought about that at all.
Yeah, obviously it's easy for me on two different slides to say this is research and this is translation, and there's going to be a blurred bit in between. You will often see basic research papers finishing off saying, and we hope these findings are relevant for interventions or translations and so forth. I think what I'm pointing to here is that different methodologies may be appropriate if you've got research versus translational aims. So the translational aims suggest that you might not want to look at these extremes, that your focus in a translational framework is to understand what the maximum variation is controlling for the genotype, because that takes you to environmental influence rather than focusing on maximizing the genetic variation, which will focus on the genetic causality. And I particularly say it's the lack of mechanism concerns me from a research perspective. You've got to do the basic research first, right? It's you build the foundations and then translation comes on the top of that. So I'm not saying you don't do these very large scale genome-wide association studies. I'm just saying in the absence of mechanism, it really undermines your ability to do the counterfactual part, to understand how things could be different, and then to get into that translational framework. So I think, it's just reflection for research. What are you trying to do here? Because pick the methods that are going to be appropriate to your goals.
Thank you. Wonderful response to definitely not a softball question. So I can see, so Guy has got quite a few questions, but before that, I can see Alex had a few comments about work that he has done in the past. I don't know if Alex wanted to say anything about that or had any questions or reflections. Or if not, we can, not really. There we go. Well, thank you for sharing your perspective anyway. In that case, I think we will move on to Gaia.
Okay.
And again, I don't know if Gaia wants to maybe unmute and just say this, or if hi Gaia, how
Hello,
are you?
Gaia.
There she is.
Hello. Apologies for the verbal diarrhea. I really enjoyed the talk. So yeah, lots of food for thought. So I wrote a couple of questions. You choose, Fred, which one you like best.
Oh, Yeah, I probably can't do reading here, so just choose
No,
the
so
hardest and give me a
well,
version.
so for the first one, and maybe we can stop at that, is you highlighted that identifying the children who have the current lowest achievement could be a really good way of narrowing the gap, because they're the children who bundle up both the highest polygenic risk and then possibly the poorest environmental trajectories so far, or maybe other developmental factors. Does that in turn mean that we need to understand and explain to educators and policymakers the trajectories to current achievement itself? It feels like, in order then for them to intervene effectively, then we have to be able to say that they're measuring achievement well and that we can all understand it well. And can your models do that as well? Could your models be modified to unpick the mechanisms of current achievement more, and how environmental interventions are more or less efficient for children of various kinds of risk?
Yeah, I mean, you're getting into the complexity of the mechanisms, which is the barrier to translation. But obviously, with my models, I jumped in at a certain point and said achievement today, and the question you're saying, how did you get to that point, and what were the influential factors? Because maybe we can intervene to go in earlier and do that. And I think, when do you start to consider best points for intervention? I talked about genetic determinism, but it's not the only branded determinism out there. There are at least two others. One of them is environmental determinism, that says if you don't change the environmental conditions, you're likely to get the same outcome. And the other one is developmental determinism, that says things get harder to change the older you get. Now, this is sort of partly sensitive period type thing, but I think it does point you to the earlier factors and trying to understand what are the key environmental factors that put you at different points on the trajectory at different ages. I know there's a lot of behavioral genetics work. This is more kind of twin study model, trying to identify at what age various genetic influences were taking place. I cannot say that my models are complicated enough to get into the huge complexity of developmental trajectories, but I certainly think diving into that causality is very important.
Amazing. Thank you so much, Kaya. And I loved all your questions. So we are just coming up to 5, but I really want to ask Elizabeth's question because it was on my mind as well, actually. So I think it's a great question to finish on. Many decades ago, I was inducted into educational research with the nature nurture discussions. Have we advanced?
Yes.
Good question, isn't it?
And I would say we have. I got into studying genetics in an educational framework because I was interested in the environmental side of it, and I thought you just can't look at one side of the coin. So you have to study both sides of it. I think we understand more of the biological mechanism in terms of brain development, the neuroscience, where genetics is working. I think we understand more about the environmental factors. I think we have more complex statistical methods to unpack how those influences are interacting. So I would certainly say we are making progress on that. I do find it sort of vaguely amusing that you could ask a person on the street, what do you think is more important in human outcomes, human variation? Is it nature? Is it nurture? A person on the street would probably say, I'm guessing, but if any of you are on the street, see whether this is what you guess. It's about half and half, right? Under normal non-extreme circumstances. There was a paper in 2015 that did a meta-analysis of all twin studies that had ever been run on any phenotype, behavioral, medical, morphological, height, weight, all of these things. 14 million participants, 3,000 traits, averaged over all of it. How much was nature and how much was nurture? And how much was nature was 49.3%. So, it's about half and half. So in that sense, not made a lot of progress, but I think we've made a huge amount of progress in the mechanisms.
Wonderful. Well, thank you again so much, Michael, for sharing your wisdom and sharing your work with us. And hopefully we will, well, I'm sure we will have you back again sometime to share another piece of your work with us. Thank you so much. So yeah, thank you, everyone. That is another CN seminar concluded. Next week, I hope to see you back again, and we will be discussing the Accessing Disability Library. So it's a wonderful new initiative from our colleagues at the University of York. So thank you, everyone. Have a great rest of your evening.
Thanks so much, everyone.
Bye.